Well, thank you for coming. I gather it's a busy time of year, so we maybe don't have as many people here as we might otherwise have, but I'm glad to see you. And today I'm going to be talking about amyloid, the protein, and I'm having trouble with my pointer already, there it is, as a biomarker of Alzheimer's disease. And the work I'm going to talk about currently is taking place in postmortem tissue, but we will be moving into live eyes, hopefully in the next few months. And so I want to talk a little bit about that, what we're going to do, what kind of instrument we're going to build to move into live eye imaging. I don't have to go back to my machine. So this is part of a theme in my lab of using the eye as a window on the brain. You've all heard, I'm sure, of the eye as a window on the soul, but in our case, we are using the fact that the eye is transparent to light in order to look at the retina, which consists of blood vessels, as you see here, and also neural cell layers. And so we expect it to act in many ways the same as the brain, with all the neurons that are distributed along the surface and in depth within the retina. So one of the other two areas of specialization in my lab is high-resolution optical imaging of the back of the eye, and that's the area that we're going to build on here in doing these experiments, the fact that we do high-resolution optical imaging and are very good at that. And I should have said in the previous slide, the other area that I work in for the interest of those of you in the audience is on the effect of the optics of the eye on myopia and its development, and how we can actually slow that myopia development down. That's not what I'll talk about today, but I thought I'd mention it anyway. So the question is, I probably can't start the movies without the machine. So let me, here. So this movie is of the retina. At a depth where we're able to see the individual cone photoreceptors, and those are the bright small spots here, and the dark shadows are blood vessels above the photoreceptors. And so this is an example of confocal imaging. How many of you are familiar with confocal microscopy? Yeah, so it's similar. We're actually optically slicing, and you can't see the vessels. They're casting a shadow on this surface, which is below the level of the vessels in the retina. And these cones are about 3 microns in diameter, and that gives you an idea of the kind of resolution that we have when we do this optical imaging. And this particular imaging is using adaptive optics, so we get higher resolution when we use adaptive optics. And we're also interested closer to the surface of the retina in using this imaging to detect blood flow. And you can see small vessels here where you saw some flow, and also the larger vessel, vein and artery. And the bright area here is actually important to us in the Alzheimer's disease. It is, in fact, the optic nerve fiber layer, and it is a layer of fibers that are coming from the ganglion cells, which are neural cells beneath this layer. All those neurons, they gather together and run along the surface of the retina towards your optic nerve. And I can take you back and remind you that you have an optic nerve, which is actually not centered on the eye. It's about 15 degrees off center. And this is where all those nerve fibers from the various ganglion cells run along the surface and then out the optic nerve to the brain. So that is definitely an area that we're interested in looking at in Alzheimer's disease, and I'll tell you next why that is. But meanwhile, just give you a normal distribution of cone photoreceptors, and beside it we have an abnormal eye condition called central serous retinopathy. And here you can see that those cone photoreceptors are no longer present, and in this even higher resolution image here, you can see that in fact it's not true that they're not present. There are in fact some present, and others are missing. And of course, the higher resolution that we use, the more eye movements cause a jumpy image during the video. But we've used this technique now to look at conditions such as this, also to look at cone photoreceptor loss in young children, and in those children being able to see if some of the cones are intact is important to the idea of stem cell therapies or gene therapies. So this kind of imaging may be married with those two technologies in curing some of those cone losses, the genetic ones. And we also look at diabetes and the first effects of diabetes on the eye, on the retina, whether it's on the vessels or on the neurons. So now I want to move to the main point of this, this talk, which is the eye as a window on the brain. And so here, this is some early work we did on cerebral malaria, and what happens is that many young children in East Africa, these tissues came from Malawi, fall into a coma, and when they arrive at the hospital, it's very important that they get treatment, the right treatment, very rapidly. But if you do a blood test, you'll find that most of them, in fact, do have malaria. The question is, do they have malaria that has affected their brains and caused them to go into coma? And so malaria starts to affect the brain after it crosses the blood-brain barrier. The blood-brain barrier is very similar to the blood-retinal barrier, and so if we can see whether or not the malaria has started to cross the blood-retinal barrier by imaging, then we'll have a very good handle on whether it's also potentially in the brain. And so that's another project that uses the eye as a window on what's happening in the brain. And this is the same kind of technique that we use in the Alzheimer's disease. What's showing up as bright spots here are the hemazoan within that the parasite produced inside the blood vessels, and hemazoan are birefringent. And what that means is that they interact with polarized light. And so this particular image is created by putting the vessels effectively between crossed polarizers. In what we do is we send light of one linear polarization, so the vibration of the E field in one direction, into the eye. It reflects back from the vessels, and we put in a crossed polarizer that then absorbs this, so the incoming polarization light was in this direction, and then we absorb this direction as things come back out, and so the only light that remains is light that has been turned from this direction to this direction by the actual hemazoan. And so that's how we use polarization. So amyloid beta is a protein that is found in the brain in the plaques of Alzheimer's disease. And we were very interested in what its polarization properties were, whether we had a chance to image it using polarized light, not using a dye. In our initial experiments, you'll see that we compare with finding of deposits using a dye. This dye is thioflavin, which labels amyloid. And here you can see a signature from polarized light imaging. So lots of funders and collaborators in this work. We have a current CHIRP grant from the Canadian Institutes of Health Research and the Natural Sciences and Engineering Research Council, also a collaboration with the Eye Bank of Canada, and lots of co-investigators needed for this work on Alzheimer's disease. And we get some research materials from Inter Vivo Solutions. So why is it important that we develop a method of imaging the presence of Alzheimer's disease by imaging the retina? Well, when we started this work back in around 2011, amyloid had been found in the retina by some investigators, but not by all investigators. In fact, there was only one report of amyloid in the retina in Alzheimer's disease in humans. The other reports had to do with animal models. And there were, though, visual changes that had been measured in Alzheimer's disease. And so when the vision is changing, you wonder if that's changes in the brain or changes in the retina. And more importantly, being able to image amyloid optically would allow us to intervene earlier in the disease, have people start on early stage therapies, and improve their quality of life. So currently there is no cure for Alzheimer's disease. The drugs that are available will help with the symptoms, and they allow people to stay independent for longer because they stop the progression of symptoms at the level usually at which the person starts the drugs. And so then for a number of years, the disease will progress more slowly, and then ultimately the person will, of course, still have difficulties and need more care. But it would give people a number of years of improved quality of life. Also, if we knew for sure it was Alzheimer's disease, that's important as well, since it's difficult to differentiate causes of dementia as to exactly what the cause is. So the other important thing is that this is essential for preventative strategies and for the development of new treatments. Because currently, when a company is developing treatments for Alzheimer's disease, they need to do multiple brain scans to see what's happening to the amyloid load in the brain. And those run $2,500 to $3,000 a scan, and so it makes any studies of new treatments for Alzheimer's disease very, very expensive. And so this would be a much less expensive method of quantifying the amyloid. So the burden of Alzheimer's disease is quite large within Canada, and it's going to go up because of the aging population worldwide. The numbers are quite staggering of people with dementia. And as I said, the definitive diagnosis is difficult among people who are living unless they have a PET scan, but PET scans are, positron emission tomography scans, are only available usually at large medical centers. And so being able to tell definitively that there is dementia and amyloid, which is the diagnosis of Alzheimer's disease, usually occurs only after death. And a lack of a proper diagnosis means many people are not getting treated at all, or they are not getting the proper treatment because the cause of their dementia is not known. So we want an early or accessible non-invasive diagnostic to maintain quality of life, and in order to allow testing of experimental treatments, and an objective testing that would allow you repeated scans and to say, yes, the amyloid load is getting better, it's getting worse. And this could then become a simple technique which would be used for screening and referral. So this is what currently happens in diabetes. Diabetes often causes vision loss, and diabetics are told to have eye exams once a year, and then as needed they're referred for better control of their diabetes to an endocrinologist. And in this case the referral would be from optometrists or ophthalmologists to neurologists for full assessment of the patient. So why polarimetry, and why did we think it would work? Because amyloid beta is known to be a fibrillar protein in the plaques in the brain, and also because for many years, starting in the 1920s, it was stained with Congo red. And when it had been stained with Congo red, it became, the stain deposit was birefringent. And between crossed polarizers, it gave an apple green color. And so that for many years back in the 30s and 40s and 50s was the classic way that pathologists actually figured out if there were plaques in the brain. And so it occurred to me that perhaps the Congo red wasn't needed. Perhaps you could pick up a polarization signal without a dye. And of course that would be much less invasive than using a dye. So our purpose was, first of all, to study the occurrence of amyloid deposits in the retina, because there was this literature, some people had found it and some people had not. And secondly, to compare the amyloid deposits in human and a canine model of Alzheimer's disease. This is a naturally occurring condition in the dogs. They become impaired at older ages, and they develop amyloid in their brains, and it's analogous to Alzheimer's disease. And of course, because of their shorter lifespan, we're able to get more tissue than from the humans, and so that helps us in developing our methods. So we wanted to study the visibility of this amyloid in different imaging modalities, and particularly imaging with polarized light. So in this particular cohort, we had 20 humans with a diagnosis of Alzheimer's disease and 22 age-matched controls with no dementia and no Alzheimer's and no glaucoma, which has also been associated with amyloid. And the retinas from the canines, at this point we had 6 positive and 7 negative for cognitive dysfunction syndrome, which is the name of the disease in the dogs. We flat mounted these retinas, and this is where we differed from many of the previous groups. Often people use a staining, an immunofluorescent staining for amyloid, and they take thin sections of tissue and do that staining. The retina is a fairly large object when you actually extract it from the eye, and so if you're doing thin sections, you're not going to sample the whole of the retina. And it occurred to us that the difference in the studies could have been that the deposits are somewhat sparse. And so we flat mounted our retinas on slides, and then we searched over the whole area of the retina. And we used initially fluorescent amyloid markers like thioflavin, and then other imaging techniques, atomic force microscopy, confocal microscopy, and polarization microscopy. And then we started our first in vivo imaging about a year ago in the canine model. So this is the confocal scanning laser microscopy, and the green fluorescence is from thioflavin, so you can see a relatively large deposit. Here's the scale. It's got a length of about 150 microns, and then a couple of smaller, 3 smaller deposits looking down upon the surface of the retina. And reconstructing the confocal sections to get a cross section, then you see the larger deposit and these 3 smaller deposits. And the blue is DAPI staining, which is a nuclear stain. And what you can see is that these deposits are sitting in or slightly above the top cellular layer of the retina. And that is the ganglion cell layer. So that is a neuronal cell layer, which is where we would expect to find amyloid in association with Alzheimer's disease. And so above it, you then still have the optic nerve fiber layer, which would not stain with DAPI. So they're embedded very close to the surface of the retina. So what did we find in terms of sensitivity and specificity when we used thioflavin fluorescence? So we had for the 20 people with AD, 17 of them showed thioflavin staining indicative of amyloid in the retina, and 3 did not. And then for those without Alzheimer's disease, 9 showed staining, and 13 did not. And this sensitivity is reasonably good, especially since eye bank tissue, the diagnoses can come from a hospital chart, but if someone dies at home, it can also come from a family member. And so it's very hard to get a diagnosis that you can say with 100% certainty is the correct diagnosis. It could be dementia that a family member has named as Alzheimer's disease, for example. And here, the 59% specificity is very interesting to us because when imaging is done in PET amyloid scans of the brain, they also get approximately 60% specificity, which means that they find amyloid in many brains of people who have no symptoms of Alzheimer's disease. And the current belief is, from the studies that have been done tracking people, that amyloid occurs in the brain 10 to 20 years before the first symptoms of the disease. And so you would, in fact, expect a relatively large number of positive amyloid measurements for people who were not yet diagnosed. And so what this number implies is that we may be seeing amyloid almost as early in the retina as you see it in the brain. That is years before the actual diagnosis. We also did atomic force microscopy. How many of you are familiar with atomic force microscopy? Yep, not too many. Okay, so in this technique, you have a cantilever that with a point on it, and it is being scanned across the surface of the retina, and it deflects if there's any change in height of the retina, and you can measure that deflection to within nanometers by using a laser beam that's reflected off the cantilever itself. And so our thought was if these deposits are really close to the surface of the retina, then we should see that deformation of the surface. And this is, again, a fairly large deposit. This is a 25 micron scale bar. And this is, again, thioflavin staining. But you can see some milder staining all around there as well. And so this is our first AFM scan of a fairly large field. And anything that is white is a large deflection relative to the things that are darker color. So you can see that over top of this stained deposit, the retina is higher than it is in the surround. And you can see these very interesting structures here also picked up by the AFM. So then when we take a high resolution scan over a smaller area, and that's the area here, then you start to see the structure of these deposits, and we can then rescan one of the deposits at very high resolution. And you can see, you probably can't see in fact, that the range of heights varies by about 2 microns. So there's some deformation of the surface that corresponds to these deposits, which again suggests they're very anterior in the retina. And interestingly, this conformation is the same conformation that is found in the brain for amyloid plaques. So some of them have this what we call a donut shape when they're found in the brain. So then back to using polarized light. And so this is an example of scratched polarizers and the 2 linear polarizers that are parallel. And then when we cross one with respect to the other, the background goes completely dark. And now we're putting, between those polarizers, we're putting magic tape, the Scotch tape that is invisible, and plastic fork, and then turning the polarizer first parallel and then crossed with respect to the first polarizer. So these samples are in between the 2 polarizers. And as we turn it to the crossed position, you can see that the area around the plastic goes black, and the plastic itself has some contrast, and certainly the tape does as well. And that's due to the birefringence of the material placed between the polarizers. How many of you know what birefringence is? Okay. So birefringence means that the speed of light through the material differs for a vibration of the electric field of the light in one direction from another direction. And so what we are measuring, what we measure in our method is the retardance in the time that it takes to go through our sample, how far behind one of those vibrations is from the other vibration. And that's what actually produces these interference colors, because the 2 vibrations are now offset from each other, and they can either interfere constructively and give bright colors, or destructively and give dark colors. So how do we do our polarimetry measurements? Well, we have modified, with the help of the company, a Nikon microscope, and we have what's referred to as a polarization state generator, a linear polarizer plus quarter wave plate, which we rotate this quarter wave plate to 4 different positions, and then polarization state analyzer, a quarter wave plate rotatable again, and a linear polarizer. And what we do is we choose 4 positions of each of these 2 polarimetry measurement and generator. And so we take 16 images of our sample. And these are images in transmission in the microscope. So we do that, and we take these images as a function of the pixel position on the retina. And so these are what are known as Mueller Matrix values for each position on the retina. Now, what a Mueller Matrix value is it's the, it tells you the interaction of the tissue with the incident polarized light. So it is a property of the tissue, and you can very clearly see that there is a deposit. This would be an image that you would see with unpolarized light. So you can see there's not a lot of contrast between the deposit and the surrounding retina. But here you can see much more contrast between the deposit and the surrounding retina. It's clearly visible in some elements of the Mueller matrix. And from these elements, we can then calculate how the deposit and the surrounding retina are interacting with the incident light, all of the ways in which the tissue interacts with incident light, and that's what makes the Mueller matrix such a strong method. And so some of the things that we can calculate from that is the degree of polarization. So if I send in light that is all in one vibrational state, it's called polarized light, and how much of it then comes out in randomly polarized directions. That would mean that the degree of polarization is reducing. And if I put in randomly polarized light and it comes out polarized, then that's referred to as polarizance. It means that you're interfering with the states of polarization in different ways. Deattenuation is dependence of transmission through the object as a function of polarization state. So it may absorb some polarizations and transmit others. And birefringence is what I referred to initially. It's a difference in the speed that the light traverses the tissue for each of the polarizations, and that's reflected by a difference in refractive index. So here's our crossed polarizer image, first of the magic tape and then of the deposit you saw the Mueller matrix for. And then here is showing that, yes, it's amyloid. It is thioflavin fluorescent. And then here are two properties calculated from the Mueller matrix. So this is the amount of depolarization. That is the amount of polarized light that is turned into unpolarized light. And that is normally associated with scattering, and so is a property you often find in tissue. And you can see that the retina has a depolarization index close to 1, which means that it doesn't depolarize very much. And the lower numbers mean that the deposit is tending to scatter light and turn polarized light into unpolarized light. So that's a property that well defines the deposit. And the other is this optical retardance, which is the phase difference between two polarizations that is caused by a difference in speed through the tissue. And you can see the surrounding retina has a value down around 20 degrees, but the value in the deposit is up as high as 160 or 180 degrees, which is quite large in terms of retardance. So you can clearly see the deposit if you look at either of these properties. And we haven't used a dye at all except to confirm that it is amyloid, but we're not actually looking at the fluorescence of the dye in these particular pictures. And we have tested that the dye itself doesn't have polarization properties. So when we then take 214 deposits across 7 retinas, and 7 control areas of retinas from people without an Alzheimer's diagnosis, so they have no deposits. Then we can see how many of those deposits that were thioflavin positive actually show polarization properties as well. And again, over 80% of them do, and very, very seldom do we find an area of the retina that shows polarization properties that isn't a retina from an Alzheimer's disease patient. Only 0.8% of the deposits or of the areas we looked at would show some kind of polarization contrast and not be from an Alzheimer's subject. And if we look just at in every retina that had AD, is there at least one deposit that shows polarization properties? The answer is yes, 100% of the time. And the specificity, the retinas that did not have AD, did we ever see an area in those retinas with polarization contrast, and the answer is never, so we have 100% specificity. This 99.2% is areas of retinas with AD that didn't stain with thioflavin but still showed polarization contrast. So here we have the same deposit again with the same two properties that you saw previously, and below it we have a deposit of pure amyloid, which we deposited on glass, and you can see that this deposit of pure amyloid has very similar polarization properties: depolarization index, deattenuation, optical retardance, and polarizance, as what we assume is an amyloid deposit within the retina. And this is a control retina, and you can see that the retinal area is much more uniform in the control retinas, and quite low values of these properties. Similar results in the dog, and I'm just wondering, I don't see a clock here, so I should get 3 o'clock. Okay, so similar results in the dog. Won't go through them in detail, but we get 100% sensitivity and 67% specificity for fluorescence. Again, this 67% means that we're seeing amyloid in the retinas before the dogs are diagnosed with cognitive impairment. But every dog diagnosed with cognitive impairment, in fact, had amyloid in the retina. And then when we do the polarization contrast, we're getting high sensitivity and high specificity for deposits in the retina. They can also then be imaged with polarization, and areas of negative for amyloid can be well separated from areas positive for amyloid. So this is the human deposit you saw previously, and this is an example of a dog deposit. And so you can see very similar footprints in each of these properties. And so what that tells us, and both of these deposits stained with thioflavin, what that tells us is that the dog deposits in the retina are a good model of what's happening in the human retina. So then, of course, we wanted to move to imaging in live eyes. And that's very important. I mean, that's where this whole project is going ultimately, to have a non-invasive live eye imaging method. And so we had available from Massachusetts General Hospital and Harvard University a new dye for amyloid that had been developed for two-photon imaging, but ought to be able to be excited with a single photon. And so this is a dog eye, a living dog, and under anesthesia, we then injected the dye. First of all, we took pictures of the retina in fluorescence mode. So this is a blue autofluorescence mode in the instrument, but we can look for fluorescence before we do the injection. And so in this particular area, you can see there was no evidence of a deposit. There's some evidence of structures that produce fluorescence without a dye being present, and we refer to that as autofluorescence, tissue autofluorescence. And then after 5 minutes after the injection, you can clearly see these punctate deposits all over the retina that correspond to areas where there were no deposits before the injection of the dye. So this is uptake by these deposits of the fluorescent dye, and followed by being able to actually image them. So this is very exciting in terms of knowing this, that we can resolve the sizes of the deposits, that they're there in a living eye in the same way they're there in a postmortem eye. But there are very few instruments we can easily modify with our polarization method. So basically, our current round of funding also allows us to build from scratch an ophthalmic imaging instrument that will do the polarization imaging. And then, of course, we'll want to compare the polarization imaging with the blue autofluorescence and dye imaging in the canine, and with imaging of the brain in the human subjects. So also, there had been some reports that optical coherence tomography was able to pick up these deposits. And so this is an example of an optical coherence tomography scan taken through a deposit with this instrument. And there's no evidence that you can see anything in this area that would correspond to a deposit without polarization imaging or a dye. And even with, and optical coherence tomography is not sensitive to the fluorescence from the dye because it is done in infrared. So you basically see no contrast in that position of the retina. There have been some people who've speculated that the deposits scatter light enough that you might see them without having to use any special method. And we feel that the answer is no, we don't see them. We checked across a large number of deposits, took the scan that appeared to go directly through them, and 10 or 20 scans either side of the deposit to make sure nothing was showing up in the position of the deposit. So, conclusions from these studies to date, amyloid deposits with similar properties are found in human and canine retinas, and that strengthens the idea of these deposits being a biomarker of Alzheimer's disease, that these amyloid deposits are occurring in conjunction with Alzheimer's disease and its equivalent disease in the dogs. And we get a specificity close to that of positron emission tomography amyloid scans, which indicates that amyloid deposits are likely present in the retina prior to disease symptoms. And this is important because many of the companies developing new Alzheimer's drugs believe that those drugs have been used to date too late in the disease, and that some of them have failed phase 3 clinical trials because there's so much amyloid present that they can't have enough of an impact. And so they have been arguing for early stage treatment of people with amyloid deposits before they have symptoms. And in fact, there are several studies now going on in Canada of that cohort of patients. If they are at high risk for the disease, either because family members have had the disease or they have a genetic marker for the disease, they're actually being allowed to enter clinical trials before they have any symptoms and test these drugs with the hope that that will mean that the symptoms will never develop. So the similarities between the deposits support the use of the canine model of Alzheimer's disease. And I hadn't talked about the subsets here, but when we look at those dogs with cognitive dysfunction syndrome, and we look at dogs who have deposits and not yet cognitive dysfunction syndrome, then we find that the deposits that happen before cognitive dysfunction syndrome are, for the most part, not as large and somewhat thinner than the deposits that occur with the syndrome. So it may be that we can use the thickness or the area of the deposits to stage the disease. That's also a hope that we have. So these deposits appear to contain a high concentration of amyloid beta because they have properties very close to the pure amyloid beta. And this is a promising non-invasive method of imaging the deposits as a biomarker, which we are hoping to make a widely available test. And as I said before, important for implementation with clinical trials. So what are our next steps? We funded to build the prototypes for polarization imaging in living humans and canines. And those studies will compare polarization markers with established biomarkers of the disease. And the market we're looking at initially is drug development and research, and then ultimately a screening method for earlier pre-symptomatic detection of biomarkers, particularly if one of the early stage drugs is successful, and in order for it to be successful, people need to be treated before they have symptoms. Then this would become a much more important biomarker of the disease. And we have a prototype instrument design chosen. We're going to take 16 images in half a second. We have the funding for the study, and the 3 Alzheimer's research groups that we're collaborating with are at Western University, UBC, and University of Montreal, and lots of funding sources needed for this kind of research. So thank you. And I'd like to open things up for questions.
Thank you for your great talk. I'm wondering if amyloid deposits, are they a symptom of the Alzheimer's disease, or are they kind of an indication of that? Can they be perceived as a cause, like cause of the disease?
So I didn't, because of the time constraints, I didn't address that question, but it is a very important question. So the current state of the amyloid hypothesis, that is the hypothesis that amyloid causes the symptoms of the disease, is under question because some of those drugs have failed. And so there are 2 theories. One is that maybe amyloid is not the fundamental cause of the impairment. And the second thought is it just needs to be treated earlier. So our approach is, as long as we find a biomarker of the disease that occurs early enough in the disease, it doesn't actually matter if that biomarker is the cause. The thought is that the most toxic thing is the early dimers that ultimately form the amyloid beta fibrils. That it's not the fibrils themselves that are the most toxic. It's the dimers, the 2-molecule combination of amyloid, that is actually most toxic to cells.
Okay, so the only concern would be methods, or like you mentioned drug developments and research. Mm-hmm. People developing methods that would solve the problem with the amyloids or completely remove them, but yet people still
question whether that will actually help the disease. That's right. But on the other hand, we know that for sure through PET scans, amyloid PET scans, that the amyloid occurs very early in the disease. So it's definitely a marker of early stage disease. The other thought that people have is that the treatments might be of 3 different things. They might be of the amyloid, they might be of the tau protein, which causes fibrillar buildup in a different way. And the third thing that they're looking at now is inflammation that occurs in the brain in Alzheimer's disease. So there might actually be sort of 3-way treatments ultimately. But nonetheless, it's a biomarker of the disease before any symptoms occur.
I'll ask one more question. It's different from the previous ones. In your dog model, the in vivo, you mentioned you inject a dye inside the eye, or is it?
No, actually we did it intravenously. Mm-hmm.
Okay. And is 5 minutes that you mentioned, is it long enough for actually
It's long enough to actually get to the eye and to fill the deposits, yeah. It's actually quite surprising, but that's a timing that was already known for dyes like fluorescein that are used to map out the blood vessels in the eye, so that's why we chose that timing, and yes, it was long enough. I mean, we don't know for sure that there aren't more deposits that it would have diffused out from the vessels towards had we left it longer. Obviously, those kinds of studies we would also like to do.
Okay, so it's possible actually to use these dyes in human as well?
This particular one has not been approved for human use. So thioflavin as a dye in human, potentially, but I don't. It's interesting, all the things I didn't know when we started this. The PET scans use a form of thioflavin that is then radionuclidide tagged, right? But the advantage of a PET scan, lots of disadvantages like cost and radiation, the advantage is that they're very sensitive, so you need a very small amount of the dye. It's likely that if you were using thioflavin as a dye marker in the brain or in the eye, that you'd need much more of it. And then I don't think its safety has been tested.
Thank you.
Any other questions? All right. I don't know. I think probably I finished a little early.
Thank you so much.
Oh, you're welcome. Thank you.